Hello students, welcome to EPG Pathshala. I am Dr. Jayant from the Department of Biochemistry at the All India Institute of Medical Sciences, New Delhi. Today we are going to discuss about the module DNA replication models and mechanisms from the paper Cellular and Molecular Biophysics. So students, let us see what we are going to learn in this module. Now in this module, at the end of this module, you should have the following objectives covered. So this includes the various models of DNA replication. Now there are multiple models of DNA replication which you should know, and you should also learn about the Michelson-Stahl experiment. Then we'll move on to prokaryotic DNA replication, the basic mechanism and the proteins involved. Then we'll move on to the higher level of DNA replication, which is eukaryotic DNA replication, the proteins involved, and what is the difference between prokaryotic as well as eukaryotic DNA replication. Finally, we look at the role of telomerase, which is a very important component of eukaryotic DNA replication. Now let's start off with the class. Now, as an introduction, DNA is the human genetic code. Now, one of the landmark experiments which established that DNA is the human genetic code was the Avery, MacLeod and McCarthy experiment. Now, this experiment was carried out at the Rockefeller Institute in 1944. Now, what was this experiment? It was a very elegant and simple experiment. So, at that point of time in 1944, two types of bacteria were known, streptococcus. So, two types of streptococcus. One of them we name them as the S type and the other were the R type. Now S and R refers to the type of colonies which they form in culture. Now these scientists already knew that the S cells usually are very virulent. So injection of the S type of bacteria into the mouse usually causes the death of the mouse. On the other hand, the R cells are not virulent, and if you inject them into a mouse, the mouse remains healthy. And if you heat kill the S bacteria, that meaning you destroy it and then inject it into the mouse remains healthy. However, the landmark experiment was performed when the heat-killed S bacteria were mixed along with the R bacteria and injected into a mouse, and the mouse died. Also, S cells were recovered from the blood of the mouse. So what does this mean? This proved that there was something in the dead S bacteria which had moved into the R bacteria which had caused the mouse to die. Now this could either be DNA or protein. However, people had already deduced at that point of time that DNA was the same in all cells. So they had a big hint from this experiment that most probably DNA is the human genetic code. And subsequently, they established with other experiments that DNA was indeed the genetic material of bacteria. DNA replication. Now, double-stranded structure of DNA was given by Watson and Crick in 1953. Now, their model was very simple. It also implied a very elegant mechanism for replication. Now, because the DNA was double-stranded, the model implied that replication would involve unwinding the DNA molecule, separating the two strands, then make a complementary copy of each strand. Now, DNA replication. In the 1940s and 50s, there were multiple models of DNA replication which were proposed. Now, three models were proposed. Now, let's look at them one by one. The conservative model. Now, you know that DNA is double-stranded. And how does it replicate? The conservative model proposed that both strands of the DNA separate and then they are copied. And when they re-anneal, one copy would be entirely old DNA while the other copy would have both strands of the new DNA. This was one of the models which was proposed. The next model which was proposed was the dispersive model. What is the dispersive model? Now here the double-stranded DNA might fragment, replicate double-stranded DNA and then reassemble. So there will be a mosaic of old and new double-stranded DNA regions in each new chromosome. Now, the third model which was proposed was the semi-conservative model. What is the semi-conservative model? Here, the DNA strands separate, a complementary strand is synthesized for each of them. At the end of the replication, the one of the parent strands then pairs with the daughter strand. So, the two new DNA molecules which are formed have one part from the parent and one part from the daughter. So, these is the semi-conservative model of DNA replication. Now, these various models of DNA replication were then experimentally studied by the landmark experiment which is called the Messelson and Stahl experiment. Now, you can see the three postulated methods of DNA replication on your left. One on the top is the semi-conservative, meaning that the progeny will have one daughter strand and one parent strand, or the conservative model which will have the daughter strands pairing with each other and the parent strands pairing with each other, and the dispersive model wherein you have a mosaic structure where it's a mixture of the daughter and the parent strands for the progeny. What did Messelson and Stahl do? The overview of what they did is shown in the right panel. So, as we all know, DNA has nitrogenous bases in its backbone. So, usually the nitrogen which is incorporated in nature is the N14. Now, experimentally, we can grow E. coli in the lab, and what Messelson and Stahl did is they grew E. coli in a medium containing N15, which is an isotope of nitrogen which is heavier. Now, N15 and N14 can be separated by ultracentrifugation using the technique of density gradient centrifugation. Now, after a few generations of growing E. coli in N15, all the DNA would now be N15. All the DNA would now be heavy nitrogen. Then Messelson and Stahl moved these bacteria into normal nitrogen containing media, and then they studied what happens in the first generation, the second generation, and so on and so forth. And then they isolated the DNA and then did a density gradient centrifugation. You can see on the panel corresponding to 0 that all the bacteria have, because they have a higher density, because they have N15, have settled at a lower level. And then in the first generation, E. coli takes 20 minutes to replicate. You can see that there is a mixture of N15 and N14. And in the second generation, you can see that slowly there is a movement of the DNA from the mixture to the pure N14 containing population. You can see that in the second generation, there is a mixture of 50% of DNA which is only N14, 50% which has both N14 and N15. This conclusively proves that the semi-conservative model of replication is present in nature. Why? Because if the conservative model was present, then in the first generation you would have seen two bands, one corresponding to the N14 and one corresponding to the N15. And if the dispersive model were true, in all generations you would have seen a mixture of N15 and 14. However, this mixture which you see in the first generation followed by the pure N14 populations which come up in the second and third generation show that DNA replication is semi-conservative. So, prokaryotic DNA replication. So, DNA replication in bacteria. So, as I already told, DNA replication has a few basic mechanisms. Because DNA is double stranded, you need to uncoil the helix. That is the first thing to do. The double stranded DNA is separated into two single strands. Then strand separation is completed by breaking of the hydrogen bonds between the complementary strands. Now, once the hydrogen bonds are broken, what happens is you create kind of a space in which all your other proteins which help in replication can enter, they can bind to the DNA and then help in replication. Now, finally, there is synthesis of the new strand using the parent strand as the template. At the end of that, you will get two daughter strands and two parent strands, which then anneal to form the new DNA molecule. Now, in bacteria, replication usually begins at a specific site. Now, this site is called the ORI. It is also known as the origin of replication. Now, DNA replication in bacteria, as I told you, starts at a particular place called the origin of replication. Now, as you can see in the figure, the origin, there is a bubble which is created at the origin which separates the double-stranded DNA into single-stranded. Now, this ends of the replication bubble are called replication forks. Now, as you can see from the figure, DNA replication is bidirectional from the origin of replication. What does that mean, bidirectional? Meaning at each replication fork you will have two strands which are being formed, one using the top strand as the template, one using the bottom strand as the template. Now, how does this separation of DNA take place at the origin? This is by a group of enzymes called DNA helicases. Their role is to unwind the two parent strands of DNA, and they form two replication forks which are Y shaped, and these are bidirectional in case of bacteria. DNA replication in bacteria utilizes a number of proteins. One of them is the helix destabilizing proteins. Now, what are helix destabilizing proteins? Now, these proteins bind to the single stranded regions of the DNA once they are separated apart. Now, what is the use of helix destabilizing proteins? Now, you know that the double stranded DNA is exactly complementary to each other. Now, if they are just separated using the helicase protein, what will happen? They can very easily go back and anneal to each other to form the double stranded moiety again, meaning they would not be in the single stranded form anymore. So how is this prevented? Because we need to prevent this, because the single stranded moiety has to serve as the template for the daughter strand. For that, there are specific proteins called helix destabilizing proteins or single strand binding proteins. What do? They go and bind to the single stranded region of the DNA and prevent them from forming a double stranded moiety again. So after the helix destabilizing proteins, there are also enzymes called topoisomerases which are important. Now, what are topoisomerases? Now, these topoisomerases produce breaks in the DNA and then they rejoin them in order to relieve the stress in the helical molecule during replication. Now, as you see from the replication fork, the strands continue to unwind in both the directions, and as new complementary strands are being produced in both directions of the replication fork by hydrogen bonding of the new incoming DNA nucleotides with those on each of the parent strands. Now, how is this being done? Now, as the double stranded DNA is broken up, is separated into single stranded moieties, incoming complementary DNTPs come and line up against the parent strand. Now, these are then joined together by 5' to 3' phosphodiester bonds. Now, this is facilitated by a protein called DNA polymerase. Now, the DNTPs line up against the parent strand by way of complementary base pairing, as we know that adenine base pairs with thymine and guanine with cytosine. These are connected by hydrogen bonds, and the phosphodiester bond is formed between the 5' phosphate of the incoming nucleotide and the 3' hydroxyl group of the last nucleotide in the DNA strand. Two phosphates are removed, and this makes the reaction exothermic, thereby making it essentially irreversible. Now, the two strands of the DNA are antiparallel. What does that mean? One strand runs in the 5' to 3' direction, while the other strand is complementary to the first strand. However, it runs in the 3' to 5' direction. So, there is one important issue with the DNA polymerase. DNA polymerase can only add nucleotides from the 5 prime to the 3 prime end, meaning it needs a free 3 prime hydroxyl group where it can incorporate fresh nucleotides. We do not have any DNA polymerase which can add nucleotides in the 5 prime end. So what happens because of that? As the replication fork opens, there will be two strands, one running in the 5 prime to 3 prime and one running in the 3 prime to 5 prime. Now what will now happen? In one of them, DNA polymerase will be able to synthesize continuously because it will be moving from the 3 prime to the 5 prime direction. So this strand is called the leading strand. So it goes continuously. However, the other strand runs from the 5 prime to 3 prime direction, and because it is opening at the fork, it has to be discontinuous. So there are discontinuous small fragments of DNA which are being replicated in the other strand because of the fact that DNA polymerase can only add nucleotides in the 3 prime end. So this strand is called the lagging strand, and all these short fragments of DNA which are replicated are called as Okazaki fragments, and Okazaki fragments are around 100 to 1000 nucleotides long in prokaryotes. Now, DNA polymerase, as I said, can add nucleotides only onto the 3 prime hydroxyl group, not at the 5 prime end. Another limitation of the DNA polymerase enzyme is that it can only add the nucleotides to the 3 prime OH group of a nucleotide in a pre-existing strand. It cannot add nucleotides de novo. So to start the synthesis of the leading strand and each fragment of the lagging strand, we have an RNA polymerase complex. Now, this complex is called primosome and the enzyme is called primase. Now, what does this do? Now, RNA polymerase has the ability to add nucleotides de novo using one of the DNA strands as the parent. So it does not need any pre-existing nucleotide. It can just go bind to the parent strand and then add ribonucleotides. So the primase is capable of joining RNA nucleotides without requiring a pre-existing strand of nucleic acid. So this forms something called an RNA primer, which is around 8 to 10 nucleotides long. Now, as I had told previously, you can look at this figure. The purple ones are the RNA primers which are being formed. These are usually 5 to 10 nucleotides long, and this is done by an enzyme called primase, which is an RNA polymerase. After the 10 nucleotides are incorporated, so these are ribonucleotides, so this is RNA, then the primase is replaced by DNA polymerase. Now DNA polymerase, now because it has a free 3 prime hydroxyl end of this short RNA primer, it will simply start adding nucleotides to it. All this stems from the fact that DNA polymerase cannot start a strand de novo. This RNA primer is later degraded and filled in with DNA. Now, this is what happens at the replication fork. As I told you, DNA polymerase 1 helps in removing the RNA primase and replacing it with DNA. And these nicks are usually sealed by this protein called DNA ligase. The main processive enzyme which is involved in DNA replication in bacteria is DNA polymerase 3, which helps in formation of the leading as well as the lagging strands. Now you can also see that the RNA primer is being formed by the primase. The helicase which is present at the replication fork helps to unwind the strands, and topoisomerases are present ahead of the replication fork and they help to remove supercoils. Now the single strand binding proteins helps to keep the two strands apart as single stranded moieties during replication. Now in E. coli there are multiple DNA polymerases. They are polymerase 1, 2 and 3. This table gives an overview of the characteristic properties of each one of them. Now you can see that all the three have 5' to 3' polymerase activity, and you can see that the turnover number is different for each of them, and the highest turnover, meaning the highest number of nucleotides added per second, is for the DNA polymerase 3. Along with the polymerase component, these polymerases also have exonuclease activity. All the 3' to 5' exonuclease activity which will help in proofreading. However, the 5' to 3' exonuclease activity is only for DNA polymerase 1. This helps in replacing the RNA primer with DNA. Now, as we know, DNA polymerase is the most important component of the replication machinery. Now, prokaryotic DNA polymerases are of three types. Let's look at one by one. Polymerase 1, 2 and 3. Now, DNA polymerase 1. This was the first DNA polymerase which was discovered by Arthur Kornberg, for which he was awarded the Nobel Prize. Now, DNA polymerase 1 is not the main processive enzyme which is involved in replication. However, it is involved in DNA repair. It has 5' to 3' polymerase activity, meaning it can add nucleotides in the 5' to 3' direction. Along with that, it also has a few subunits which have 3' to 5' as well as 5' to 3' exonuclease activity. What does that mean? This means that the DNA polymerase 1 has the 3' to 5' exonuclease activity which is responsible for proofreading activity. At the same time, it also has 5' to 3' exonuclease activity which is very important to remove the RNA primers which were initially formed during replication so that they can be replaced by DNA. So this is the function of DNA polymerase 1. DNA polymerase 2. This is also involved in the repair of damaged DNA. It has 3' to 5' exonuclease activity, 5' to 3' polymerase activity. However, it does not have any 5' to 3' exonuclease activity. Now, there are multiple proofs that DNA polymerase 2 is also not the main polymerase. What are the proofs? Now, there are three important points. These are all experimental proofs. The first point, knockout strains of E. coli which do not have DNA polymerase 2, they show no defect in any growth or replication. So this kind of proves that in the absence of DNA polymerase 2, the cells can live and they can replicate. So this is not the main processive enzyme for DNA replication. Point number 2, polymerase 2 is induced usually during the stationary phase of cell growth. However, during this phase, there is no DNA synthesis or replication. So, DNA polymerase 2 is most probably involved in DNA repair and not the main processive enzyme in replication. Finally, although DNA polymerase 2 has a very low error rate, it is very slow to be of any use in normal DNA synthesis. Finally, scientists discovered the main replicating polymerase in bacteria, which is DNA polymerase 3. Now, it also has a 3' to 5' exonuclease activity which helps in proofreading. It also has 5' to 3' polymerase activity, and this is the principal replicative enzyme. Now, how do we know that this is the principal replicative enzyme? Because it is highly processive and at the same time it catalyzes polymerization at a very high rate. Now let's look a little bit in detail about the structure of DNA polymerase 3. Now if you look at the picture, you can see that there are two forms of this enzyme. One is the core enzyme and the other is the holo enzyme. Now what do you mean by core enzyme? Now core enzyme consists only of those subunits which are required for the basic underlying enzymatic activity. So that will refer to the alpha, epsilon and the theta subunits. Now what do? They are responsible only for the polymerase activity and the proofreading exonuclease activity of the polymerase. That's it. So that is what the core enzyme does. What does the holoenzyme mean? Now holoenzyme refers to all this, the theta, epsilon, alpha, as well as the components which are seen in the lower part of the figure, which includes the beta sliding clamp and the clamp loading complex. Now what is this clamp complex? Now this is one important component of DNA replication. Now the beta clamp is a structure which slides through the DNA in front of the DNA polymerase. Now the loading of this clamp takes place by the clamp loading complex. Now what is the role of this clamp? As you can see from the figure, the clamp can be seen right in front of the core DNA polymerase. Now the interaction between the clamp and the polymerase is very strong than the direct interaction between the polymerase and the template DNA strand. So because of this, what happens? The clamp increases the number of nucleotides that the polymerase can add to the strand before dissociation, meaning the clamp increases processivity of the DNA polymerase. In the absence of this beta clamp, the DNA polymerase tends to polymerase only a few nucleotides before it tends to fall off. The addition of this beta clamp holds the polymerase and the template together so that the rate of DNA synthesis increases by around 1000 fold. Now, DNA topoisomerases. Now, these are special enzymes which help in DNA replication. What do they do? As the two strands of the template DNA unwind, the DNA which is ahead of the replication fork is usually forced to rotate in the opposite direction. Now, this causes supercoils. Now, if too much of supercoiling is present, just as the supercoiling you see in a telephone wire, they can stall your replication fork and then replication can stop. In order to prevent this, if there is too much of tension caused by the supercoils, there are enzymes called topoisomerases which help to relieve it. Now the principle behind the working of DNA polymerases is very simple. There are two types of DNA topoisomerases, type 1 and type 2. As you can see from the figure, type 1 makes a single stranded nick, relieves tension and then reseals the nick. Now if the supercoil is big, then type 2 topoisomerases usually come into play. They create a double stranded nick, relieve the tension so that the two strands of DNA can rotate freely and then reseals the nick. These help in relieving supercoils and stress ahead of the replication fork. Now let's have an overview of all the proteins which are involved in DNA replication. As I have already told you, DNA polymerase 1 and 3 are essential for replication. 3 is the main processive enzyme which adds nucleotides to the 3' end, and DNA polymerase 1 helps to remove the RNA primer at the end of replication. Now there are also multiple other proteins which are involved, like initiator protein which is coded by the DNA, A gene, and other proteins like IHF and FIS which also bind to the origin of replication. Now there is the helicase which initially helps to separate the double stranded DNA into single stranded. Then there is the primase which makes the RNA primer to extend by the DNA polymerase 3. We also have the single strand binding proteins which bind to the unwound single stranded arm of the replication forks and prevent them from re-annealing. DNA ligase helps to seal the gaps formed by the DNA polymerase 1 after removal of the RNA primer. Now DNA gyrase is a type 2 topoisomerase which is helpful in relieving the stress by supercoils. There are other proteins called TER and TUS proteins which help to terminate the replication after the entire genetic component is replicated. Now let's look at the various steps in replication. Starting off with the initiation of replication. Now this is in prokaryotes. Now replication starts when the DNA at the origin of replication denatures to expose the bases, thereby creating a bubble which will form a replication fork. Now, how does this occur? Now, this origin has a specific sequence. It has a minimal sequence of around 245 base pairs. Along with that, it also has 3 copies of a 13 base pair AT rich sequence and 4 copies of a 9 base pair sequence. What is the importance of the AT rich sequence? As you know, AT sequences usually have a lower TM when compared to the GC sequences in the DNA. So this minimal sequence of 245 base pairs is where the initiator proteins attach, and after that they tend to melt this DNA into single stranded form. And because it is GC poor, it has a lower TM and it is much easier to separate than the normal DNA which is seen in the other parts of the E. coli genome. DNA helicase. Now, DNA helicase comes from the DNA B gene in E. coli. This binds to the initiator proteins on the DNA and it denatures the AT rich region which was referred to in the previous slide. It uses ATP as the energy source. Now, this is the first thing which occurs during DNA replication. Now, once the helicase binds and it has separated the two strands to form a single stranded region, then comes the DNA primase. Now, this comes from the DNA G gene in E. coli. Now, what does primase do? Now, as I already told you, the DNA polymerase cannot start the synthesis of a new strand de novo. It has to be done by a DNA primase, which is a kind of RNA polymerase. Now, this DNA primase binds helicase to form a primosome, which synthesizes a very short RNA primer, 5 to 10 nucleotides. Then the work is taken over by the DNA polymerase 3. Now, as I had shown in the previous slide, you can see that this is a small depiction of the origin of replication with the AT rich sequences. The initiator proteins bind to the 245 base pair segment which is present near the origin of replication, and once they bind they recruit these helicases which then unwind the double stranded DNA into single strands, and this paves the way for your primase to come and start the replication. Now, in the figure on the right, you can also see the single strand binding proteins which help to keep the two strands apart so that they can serve as templates during the DNA replication. Now, how does DNA replication terminate? Till now, we have seen how DNA replication proceeds. Now, how does it terminate? This is an area of intense research till now. Now, what we know is that there are certain terminator sequences which are present on both sides of the replication, meaning that the replication fork extends on both sides. So, there will be a clockwise replication fork trap and a counterclockwise trap, meaning there are sequences which are present in the clockwise direction and the counterclockwise direction. They will not allow the replication fork to move backwards once they move beyond these termination sequences. Now, how does the replication actually terminate? The replication forks move beyond these terminator sequences, and once they encounter the other replication fork, it automatically stops. The replication forks stall and replication stops. Now, if you can see the figure on the right, you can see that at the end of replication, the DNA strands are usually concatenated. So, these are separated by an enzyme called DNA topoisomerase 4, which is a type 2 topoisomerase. So, it will cause double stranded breaks and then separate the chromosomes and then ligate them back together. Now, DNA replication exhibits an extremely high degree of fidelity. What does that mean? Meaning, the mistakes during the replication process are extremely rare. How rare? Now, DNA polymerase 3 makes only one mistake per 10 raised to the power 8 bases made, which is high fidelity. Now, what is the importance of this? The importance is that because this is going to be transmitted to the next generation, it has to be extremely foolproof. Otherwise, it might cause some detriments to the progeny which is being formed. So, there exist few proofreading mechanisms which take this into account. Now, what are these proofreading mechanisms in place? There are three important proofreading mechanisms which you should know. One is the instability of the mismatched pairs. Two is the configuration of the DNA polymerase active site itself. And finally, there is the inherent proofreading function of your DNA polymerase. Let's look at them one by one. The first one, the instability of the mismatched pairs. Now, inherently, the complementary base pairs have much higher stability than the mismatched pairs. What does that mean? As you all know that adenine pairs with thymine and guanine with cytosine in double stranded DNA. Now, only this pairing has a very high stability. Mismatched pairs usually have a much lesser amount of stability when compared to the perfect base pairing. So, this accounts for part of the fidelity of the replication process. This instability of mismatched pairs accounts for an error rate of 1 per 1000 nucleotides. Moving on to the configuration of the DNA polymerase active site. Now, this is a characteristic feature of the active site of the polymerase. It will only catalyze bond formation between matched pairs. It will not catalyze bond formation between mismatched pairs of nucleotides which have come to the active sites. This is also from the structure of the active site. This is an induced fit phenomenon, and this decreases the error rate to a range of 1 in 100,000 to 1 million. Now, the third and important component which is essential for the high fidelity of DNA polymerase is its proofreading function. Now, how does it work? Now, as I already told, DNA polymerase 3' to 5' exonuclease activity. Now, this is present in a different domain of the DNA polymerase. You can see from the figure that there is a small pocket which is the 3' to 5' exonuclease pocket. If there is a mismatched nucleotide which is incorporated, it is immediately moved from the polymerase site to the exonuclease site, and then it is chopped off by the exonuclease activity, and then it changes direction and it resumes DNA synthesis in the 5' to 3' direction. So, all these three important points are the reason why DNA replication has a very high fidelity. Let's move on to DNA replication in eukaryotes. Now, what is the difference between DNA replication in eukaryotes and prokaryotes? Eukaryotes have multiple origins of replication. For example, the human genome alone has 30,000 origins of replication, whereas there is only one in case of prokaryotes. And each origin produces two replication forks and they move in the opposite direction. So, because of the enormity of the eukaryotic genome, there are multiple origins of replication for DNA replication. Now, coming on to eukaryotic DNA polymerases. Now, eukaryotic DNA polymerases are much more complicated than prokaryotic DNA polymerases. Around 15 eukaryotic DNA polymerases are known to us now. However, not all of them are well characterized. Only 5 of them are well characterized. Now, they are named as DNA polymerase alpha, beta, gamma, delta, and epsilon. Now, let's look at the functions of each of them. DNA polymerase alpha. Now, this acts as a primase, meaning this also has some RNA polymerase activity. So, this helps in synthesizing the RNA primer which is required to initiate the replication. So, the role of the primase in prokaryotes is taken over by DNA polymerase alpha. Now, DNA polymerase beta, this helps in repair of DNA, base excision repair, gap filling, all the different types of DNA repair are carried out by DNA polymerase beta. DNA polymerase gamma. Now, eukaryotes have a special organelle called the mitochondria. Now, the mitochondria has its own genome. So the replication of this mitochondrial DNA and the repair is taken over by a special polymerase called DNA polymerase gamma. It also has the proofreading 3' to 5' exonuclease activity. Now moving on to the DNA polymerase delta. Now this is highly processive, has 3' to 5' exonuclease activity, and this is responsible for the replication of the leading and the lagging strands of eukaryotes. The function of DNA polymerase 3 is taken over by DNA polymerase delta in eukaryotes. DNA polymerase epsilon. Now this is also highly processive, has proofreading 3' to 5' exonuclease activity. This is responsible for DNA repair. It was also speculated that because of its high processivity, this DNA polymerase epsilon might also help in the regular replication. However, research is still on and we do not know the exact role of DNA polymerase epsilon in regular DNA replication. Now, this is a table which is an overview on the different types of eukaryotic DNA polymerases with their location. As you can see, DNA polymerase gamma, which does the mitochondrial replication, is present in the mitochondria. Now, the interesting part which you have to understand here is that eukaryotic DNA polymerases, not all of them have 3' to 5' exonuclease activity. For example, DNA polymerase alpha, beta do not have the exonuclease activity. So, they do not have this proofreading ability. So, DNA, the primase activity or the RNA polymerase activity is present only in DNA polymerase alpha, and you can see the processivity and fidelity, these are very high for delta, epsilon and the gamma. Now DNA polymerase alpha, delta are involved in replication and gamma is involved in the replication of mitochondrial DNA. Now beta is involved in DNA repair. Another important feature of DNA replication in eukaryotes is that it is synchronized to the cell cycle. Now, as you can see from this figure, there are proteins called origin recognition complexes which are bound to the origin of the replication in eukaryotes. They are usually bound by a protein called CDC6, which prevents the ORC from unwinding the DNA strand during the G1 phase. Once the cells move into the S phase, then the cyclin dependent kinases phosphorylate the CDC6, which is then removed. Then they also activate the MCMs. Now MCM stands for mini chromosome maintenance. Phosphorylated MCMs usually bind to the origin recognition complex proteins and then unwind the DNA, paving the way for DNA replication. You can see that all of this is facilitated by the cyclin dependent kinases, thereby synchronizing it with the cell cycle very nicely. Now this is an overview of all the proteins which are involved in DNA replication in eukaryotes. Most of them are similar to what we have seen in prokaryotes. You can see the topoisomerase, you can see the single strand binding proteins, the helicase, the primase, which is the DNA polymerase alpha here, the DNA ligase which helps to seal nicks, then there is DNA polymerase delta which forms both the leading as well as the lagging strands. So what is difference between replication in prokaryotes and eukaryotes? So the proteins are slightly different. For example, helicase is replaced by MCM proteins in eukaryotes. The primase is replaced by DNA polymerase alpha in eukaryotes. In prokaryotes, it is the SSB proteins, which is the RPA proteins, the replication protein A in eukaryotes. The beta clamps function is taken over by the PCNA or the proliferating cellular nuclear antigen in eukaryotes. The gamma complex or the clamp loading complex is the work of that is done by the RFC, replication factor C in eukaryotes. Now DNA polymerase 1 removes the RNA primers in case of prokaryotes. However, this work is done by a special protein called FEN or flapped endonuclease in eukaryotes. Also, in prokaryotes, replication proceeds at around 1000 nucleotides per second and it is done in no more than 40 minutes. However, in eukaryotes, it's much slower because the fidelity has to be much higher. It's only at 50 nucleotides per second. However, because there are so many origins of replication, it is completed in 60 minutes. Now, this difference is between prokaryotic and eukaryotic replication. Prokaryotes have 5 polymerases, DNA polymerase 1, 2, 3, 4 and 5. 4 and 5 are not well characterized. Eukaryotes have around 15, 5 of them are well characterized: alpha, beta, gamma, delta and epsilon. And in prokaryotes, DNA polymerase 1 is involved in the synthesis, proofreading, repair as well as in the removal of RNA primers. 2 is a repair enzyme, whereas 3 is the main polymerizing enzyme. Whereas in eukaryotes, alpha is the polymerizing enzyme and it helps in forming the RNA primer. DNA polymerase beta is involved in repair, gamma is involved in mitochondrial DNA synthesis, and delta is the main polymerizing enzyme. Epsilon, although the functions are unknown, it is well characterized, the processivity and all the other characteristics. In prokaryotes, most of the polymerases are exonucleases. However, in eukaryotes, not all of them are exonucleases. Only one origin of replication is present in prokaryotes, whereas several origins of replication are present in eukaryotes. Okazaki fragments are very long in prokaryotes, 1000 to 2000 residues. However, in eukaryotes, they are only 150 to 200 residues long. No proteins are complex to the DNA in prokaryotes, whereas histones are complex to DNA in eukaryotes, which adds another layer to the gene expression regulation. Now, telomerase. Now, telomerase is a unique phenomenon which is found only in eukaryotes and in some other organisms. Now, what is telomerase? If you remember that Okazaki fragments are formed on the lagging strand and later the primer of the Okazaki fragments is replaced by DNA. However, if you remember, towards the end of the chromosome, the last Okazaki fragment will actually not have any template ahead of it, so that you can have a DNA polymerase to replace the RNA primer by a DNA. So what happens? So you have an overhang at the ends of the chromosomes, which is depicted in the figure. Now, overhangs are usually good substrates for DNA degrading enzymes like exonucleases. So what will happen if you have overhangs? These overhangs will be chewed up by your exonucleases and slowly there will be loss of genetic material. This can again lead to a detrimental effect on the progeny when the DNA is being transmitted. So for that, nature has devised a method to prevent this kind of genetic loss from the end of the chromosomes. So uh, eukaryotic cells have an enzyme called telomerase. Now, telomerase is a ribonucleoprotein which usually adds a specific repeat sequence to the 3' end of the chromosomes. These are called telomeres. A telomere is a region of repetitive sequences at each end of the eukaryotic chromosome. Now, how does the telomere work? The telomere is a kind of reverse transcriptase which carries its own template. This template is usually complementary to the sequences which are present at the end of the chromosome. So, the telomerase enzyme usually finds the end of the chromosome, binds to it and starts adding nucleotides. What does the reverse transcriptase do? It is RNA dependent DNA polymerase. So once the complementary region has bound, the telomerase starts adding fresh DNA nucleotides and then the strand is extended. Once the strand is elongated far enough, then once again DNA polymerase can identify this and produce more Okazaki fragments. Ultimately, a lot of telomeres are added to the ends of the chromosomes so that they are protected from this damage by the exonucleases. However, the last Okazaki fragment still remains as an overhang, but because you have added so much of stuffer sequence or filler sequence, so the main genetic material is protected from damage. Now, this telomerase enzyme is highly active in normal stem cells, and again in a lot of cancer cells. It is less or totally inactive in somatic cells. So students, this is the summary of this class. So these are the take home points. So you should know that DNA replication is semi-conservative in nature. It is carried out by multiple proteins in prokaryotes, the most important of which is DNA polymerase 3. The beta clamp helps in increasing the processivity of DNA polymerase. Now, only one origin of replication is present in the prokaryotes, and multiple origins of replication are present in eukaryotes. Now, DNA polymerase can add nucleotides only in the 5' to 3' direction. Hence, two different types of replication take place in the two strands. One is the leading strand and one is called the lagging strand. In eukaryotes, DNA replication is highly synchronized to the cell cycle. Termination sequences and DNA topoisomerase help in the termination of replication. And one special enzyme which exists in eukaryotes is telomerase. It is a reverse transcriptase which helps to protect the ends of the chromosomes, and it carries its own template. Thank you.